Chapter 18, Tumor Immunology. Overview of tumor immunity. We'll look at tumor antigens, neoantigens, antigen encoded by mutated genes, antigen of oncogenic viruses, overexpressed cellular proteins, other antigen for tumor, immune response to tumor, the T lymphocyte, antibodies, natural killer cells, macrophage, roles of innate and adaptive immune in promoting tumor growth, evasion of immune response by tumor, molecularly cells that inhibit immune response to tumor, loss of tumor antigen expression. Then we'll look at immunotherapies for tumor, passive immunotherapy with monoclonal antibodies or antibody-like molecules, adaptive cellular therapy with the anti-tumor T cells, immune checkpoint blockage, targeting T cells, immediate pathway, vaccination with tumor antigens, and other approach for stimulating anti-tumor immunity. Then we look on summaries. So cancer is a major health problem worldwide and one of the most important causes of the morbidity and mortality in children and adults. The lethality of malignant tumor is due to their uncontrolled growth and spread within normal tissue, causing damage and functional impairment. The malignant phenotype of cancer results from defective regulation of cell proliferation, resistance of tumor cells to apoptotic death, and ability of this tumor cell to invade host tissue and metastasize to distant sites. In addition, reflecting our improved understanding of immune response against cancer and the therapeutic success of cancer immunotherapy, we now include the ability of tumor cells to invade host immune defense mechanism as one of the hallmark features of the cancer. The concept of immune surveillance of cancer, which was proposed by Macfarlane Barnett and Lewis Thomas in 1950s, states that a physiologic function of immune system is to recognize and destroy clones of transformed cells before they grow into tumor and kill tumor cells they are formed. The existence of immune surveillance has been demonstrated by increasing incidence of some types of tumor in immunocompromised experimental animal and humans. More recently, we have learned that immune response against many human cancer are effective, but they can be therapeutically stimulated to destroy tumors. In this chapter, we will describe the types of antigens that are expressed by malignant tumor, how the immune system recognizes and responds to these antigens, how tumor evade the host's immune system, and the application of immunologic approach to the treatment of the cancer. Overview of tumor immunity. Several characteristics of tumor antigens and immune response to tumor are fundamental to an understanding of tumor immunity and for development of strategy for cancer immunotherapy. Tumor antigen stimulate specific adaptive immune response that can prevent or limit the growth and spread of the tumor. Classical studies, pathological analysis of tumor, and animal experiments have all established that although tumor cells are derived from the host cell, the tumor elicits immune response in their host. Most evidence indicate that the clinically relevant immune response involve T cells and especially CD8 cytotoxic T lymphocytes. Histopathological studies show that many tumors are surrounded by mononuclear cell infiltrate composed of T lymphocyte and macrophage and that activated lymphocyte and macrophage are present in lymph nodes, draining the site of tumor growth. Quantitative analysis of these infiltrate in colon cancer and some other tumor types have revealed that higher number of T cells, in particular CD8 T cell, cytotoxic T cells, and memory cells and CD4 T helper 1 cells are associated with a better prognosis than tumor uh, with fewer of these cells. So this is just a diagram, figure 18.1, lymphocytic inflammation associated with tumor. Certain tumor types frequently have associated lymphocyte infiltrate, including medullary, breast cancer, and malignant melanoma. A red arrow indicates malignant cells, and yellow arrow indicates lymphocyte inflammatory infiltrates. Immunohistologically, staining of resected tumor can be used to enumerate different types of T cells associated with the tumor, such as infiltrate of CD8 T cells in a colonic carcinoma. The tumor cell appear blue and CD8 T cells appear brown. Increased density in this way is associated with longer disease-free survival. The first experimental demonstration that tumor can induce protective immune response came from the studies of transplanted tumor performed in 1950s. A sarcoma may be induced in an inbred mouse by painting its skin with a chemical carcinogen methylchloroethylene (MCA). If MCA-induced tumor is excised and transplanted into other synergic mice, the tumor grows. In contrast, if cells from the original tumor can be transplanted back to the original host, the mouse rejects this transplant and no tumor grows. The same mouse that has become immune to its own tumor does not reject MCA-induced tumor produced in other mice which have different MCA-induced mutation and expresses different tumor antigens. Furthermore, transfer of T cells from the tumor-bearing animal to a tumor-free animal can impact protective immunity against that tumor. Thus, immune response to this tumor exhibit a defining characteristics of adaptive immunity, that is, specificity, memory, and a key role of lymphocyte. Subsequent work showed that the frequency of spontaneous or MCA-induced tumor in genetically immunodeficient mice is increased compared with the immunogenetically normal mice, further establishing a role of the immune system in tumor immune surveillance. Immunodeficient humans, such as patients with acquired immunodeficiency syndrome and transplant recipients given immunosuppressive drugs, are at increased immunodeficient humans, such as patients with acquired immunodeficiency syndrome or transplant recipient given immunosuppressive drugs, are at increasing risk of developing tumor, some of which are caused by viruses reflecting defect antiviral immunity, but also some tumor not known to have a viral etiology. Immune response frequently fail to prevent the growth of tumor. There may be several reasons why anti-tumor immunity is unable to eradicate cancers. First, many tumors have developed specialized mechanisms for subverting host immune response. We will return to this inhibitory mechanism later in the chapter. Second, tumor cells lose the expression of antigens that may be recognized by the host immune system. Even tumor that do elicit effective immune response may become less immunogenic over time because subclonal that do not express immunogenic antigen have a selective survival advantage. Third, the rapid growth and spread of tumor may overwhelm the capacity of the immune system to effectively control the tumor, which require that all the malignant cells be eliminated. Ineffective immune response to cancer can be overcome by therapeutic strategy that stimulate such response, such as anti-tumor T cells can be activated to effectively kill tumor cells. As we'll discuss later in this chapter, this realization has spurred new direction in cancer immunotherapy, in which augmentation of the host anti-tumor response is the goal of treatment. The existence of specific anti-tumor immunity implies that tumor must express antigens that are recognized as foreign by the host. The nature and significance of these antigens are described next. So this was the diagram that I didn't want to not to read, that we read together. Tumor neoantigens. Normal cell display a self-peptide on MSC molecule, but there is no T cell response due to mechanism of self-tolerance. Normal cells, that's normal cells. So normal cell peptide display the MSC, no responding to T cell due to tolerance. Tumor cell will express mutation generated neoepitope new TCR contact residue to T cell lymphocytes. Then that one can happen, or peptide from the protein encoded by an oncogene virus T cell response. Let's continue. Tumor antigens. Immune response to malignant tumors are targeted to various types of molecules that cancer cells express and may be recognized by immune system. Protein antigen that stimulate T cells response are the most important types of antigen for protective anti-tumor immunity. In the past, the term tumor antigens has been used to encompass many different molecules expressed by tumor cells that are detected by binding of anti-tumor antibodies, whether or not they stimulate protective immune response. The tumor antigen that elicits T cells immune response can be classified into several groups. Neoantigens, antigen encoded by mutated genes. Tumor neoantigens are proteins encoded by mutated genes, which appear foreign to immune system because they do not exist in normal cells and new arise as cancer develops. Usually, these neoantigens are encoded by gene-carrying passenger mutation, which are the point mutation or deletion that are related to development or malignant phenotype of tumor. The occurrence of the passenger mutations reflect the genomic instability of the cancer cells. Alternatively, small number of the neoantigens may be encoded by driver mutations in tumor-promoting oncogenes or tumor suppressor genes. Because T cells recognize only peptide bound to major histocompatibility complex molecule, tumor neoantigen can be recognized only if the peptide carrying the mutated amino acid sequence can bind to the patient MSC alleles. Exome sequencing of many cancers have revealed large number of passenger mutation, and computer algorithms have been used to predict which of these mutations occur within peptide sequence that are likely to bind to cancer patients' MSC allele. Studies of cancer patients' T cell indicate that the tumor neoantigens peptides predicted to bind to MHC molecule in individual patients do in fact stimulate T cell response in those patients, and that the number of different tumor antigen specific T cell clones that are activated relates with number of mutation in the cancer. An example of this is the subset of cancer patients whose tumor have mutation in genes encoding DNA mismatch repair protein, which result in the very high mutational burden. These patients have strong T cells response to their tumor and mostly likely to benefit from therapies designed to activate T cell, discussed later in the chapter. Antigen of oncogene viruses. The product of oncogenic virus function as tumor antigen and elicit a specific T cell response that may serve to eradicate virus-induced tumor. Viruses are implicated in development of variety of tumor in humans and experimental animals. Example in human include Epstein-Barr virus, which is associated with B cell lymphoma and nasopharyngeal carcinoma, and human papilloma virus, which is associated with carcinoma of the uterine cervix, oropharynx, and other sites. In most of these DNA viruses induced tumor, viral DNA integrate in the host DNA, and virus encoded protein antigen are found in nucleus, cytoplasm, and plasma membrane of the tumor cell. These endogenously synthesized viral protein can be processed and presented by MHC molecules on tumor cell surface. Some viruses such as hepatitis B and C are associated with cancer but are not directly oncogenic. It is thought that they promote tumor by inducing chronic inflammation reaction in which tumor promoting growth factor and other signals are generated. The tumor cells may contain viral antigens, but this is highly variable. The ability of adaptive immunity to prevent the growth of DNA virus-induced tumor has been established by many observations. For instance, Epstein-Barr virus-associated lymphomas and HPV-associated cervical cancer arise more frequently in immunocompromised individuals, such as allographic recipients receiving immunosuppressive therapy and patients with AIDS. The efficacy of virus-specific adaptive immunity to prevent tumor may be due to in large part to preventing infection and eliminating infected cells before cancer develop. Vaccination to prevent infection by these viruses also decreases the incidence of virus-associated cancer. A vaccine against HPV has reduced the incidence of cervical cancer and other HPV-associated lesions. The vaccine is composed of recombinant HPV capsid proteins from the most commonly oncogenic strain of HPV, which form viruses like particle-free or viral genomes. Vaccination against hepatitis B virus has reduced the incidence of hepatitis B-associated liver cancer. Overexpressed cellular proteins. Some tumor antigens are the products of genes that are silenced in normal cells and depressed in tumor cells, or are protein made by normal cell but produced in excess amount by the tumor. These antigens are not inherently foreign for the host, but nevertheless, they stimulate immune responses. There are several possible explanations for their immunogenicity. Normally, the antigen may be expressed for a limited time or at a particular location, for example, only during embryonic development or only in tissue that are not accessible to the immune system, so there is no long-lived immunological tolerance to these proteins. Expression in a tumor later in life or in locations that are not protected from immune cells may be enough to stimulate immune response. The amount of antigen produced in the patient with cancer may be abnormally high because of overexpressions in each tumor cells and an abundance of tumor cells, and these two may be enough to elicit an active immune response. Major categories of unmutated tumor antigen that are more abundant in tumor than in normal tissue include cancer testis antigens, protein encoded by amplified genes, and tissue differentiation antigens. The expression of only some of these structural and related tumor antigen is sufficiently different from expression in normal cells to stimulate protective immunity in patients. However, many of these tumor antigens are target of antibody therapy and potential candidate for tumor vaccines. Cancer test antigen are protein expressed in gametes and trophoblastics and in many types of cancer but not in normal somatic tissue. The first cancer testis antigen identified were melanoma-associated antigens. They are expressed in the melanomas and many other types of tumor and in normal testes, Subsequently, several other unrelated genes family have been identified that encode antigen expressed by melanoma cells and are recognized by cytotoxic T lymphocyte clone derived from the melanoma patients. The MAG, melanoma-associated antigens, proteins, and these other melanoma antigen are silent in most normal tissue, except the testes and placenta trophoblastics, but they are expressed in a variety of malignant tumor. More than 200 cancer testis genes in over 40 different gene family have been identified. About half are encoded by genes on the X chromosomes, and rest are distributed on other chromosomes. It has been postulated that in most somatic cells, the gene encoding these proteins are silenced by epigenetic mechanisms such as methylation of the promoter regions, but the loci are demyelinated in cancer cells, allowing the gene to be expressed. Some proteins are expressed at abnormally high level in tumor cells because the gene encoding these proteins are amplified. One example of such a protein is the oncogenic epidermal growth factor variant called HER2 or NEW, which is overexpressed in some breast cancer. There is no evidence that this protein elicits protective immune response in patients, presumably because it is present in normal cell and induces tolerance. A monoclonal antibody targeting HER2 is used to treat patients whose tumors show high HER2 expression. Differentiation antigen are found on tumor cells and on the cell types of origin of tumor, but not on cells from other tissue. Two examples of such differentiation antigen in melanomas are tyrosinase, an enzyme involved in melanin biosynthesis, and MAT1, melanin A protein required for melanosomes function. Both CD8 cytotoxic T cells and CD4 helper T cell response specificity for tyrosine and MAT peptide are found in patients with melanoma, perhaps because these antigens are expressed at high level due to the large number of tumor cells. However, in many cases, differentiation antigen do not induce immune response because they are normal self-antigen. Even in this situation, differentiation antigen are important in oncology because they aid in accurate diagnosis of the tumor types and serve as target for passive immunotherapy. For example, some lymphomas and leukemias arise from the B cells and express surface markers characterized at this lineage, such as CD19, CD20, antibody and T cell therapy targeting against these proteins are used to treat cancer. So this is figure 18.3. Unmutated tumor antigen protein that are not mutated but are expressed more abundantly by tumor than normal cells may induce T cell response in their host. Many of these tumor antigens include proteins such as cancer testis antigens, encoded by genes that are normally not expressed in the most cells of adults because of epigenetic suppression, but they are depressed in tumor cells. Some tumor antigen may be overexpressed because of gene amplification, such as HER2, which is abundant in many breast carcinoma. Tissue-specific antigen are protein expressed by both cancer cells. Those are diagram. The normal cell types from which tumor are derived, such as tyrosinase, made by both melanocyte and malignant melanoma cells, because of their gene deregulations or the abundance of the tumor cell, the amount of this protein is high in tumor, leading to T cell response. Other antigen of the tumor. Many attempts have been made to detect the antigen in tumor cells and in the plasma of the patient with cancer by producing antibody against tumor and using these as screening reagents. Several classes of tumor antigen have been identified by this approach. It is, however, now clear that most of these antigens are produced even in normal cells, especially under the condition of tissue injury and inflammation. Therefore, the role of this antigen in tumor immunity is uncertain. Oncofetal antigen. Oncofetal antigen were the name given to protein thought to be expressed at high level in cancer cells and in fetal, but not in adult tissue. However, the expression in adults is not limited to tumor, but is increased in tissue and in circulation in various inflammatory conditions. And the antigen are found in small quantities, even in normal adult tissue. There is also no evidence that oncofetal antigens are inducer of anti-tumor immunity. Thus, their usefulness as tumor marker, target of antibody, or vaccine candidate is limited. The two most studied oncofetal antigen are carcinoembryonic antigen (CEA) and alpha fetoprotein. CEA, which is equal to CD6060, is highly glycosylated membrane protein that function as an intercellular adhesion molecules. High CEA expression is normally restricted to cells in the gut, pancreas, and liver during the first 2 trimesters of the gestation. Its expression is increased in many carcinoma of the colon, pancreas, stomach, and breast, and serum levels are also increased in these patients. Serum CEA can, however, be elevated in the setting of non-neoplastic tissue such as chronic inflammatory condition for the intestine or liver, so it is limited clinical utility. AFP is circulating glycoprotein normally synthesized and secreted by yolk sac and liver in fetal life. Fetal serum concentration can be as high as 2 to 3 mg per ml, but serum concentration in adults are low. Serum level of AFP can be elevated in patients with hepatocellular carcinoma, germ cell tumor, and occasionally gastric or pancreatic cancer. An elevated serum AFP level is sometimes used as indicator of advanced liver or germ cell tumor or in recurrence of this tumor after treatment. Altered glycolipid and glycoprotein antigen. Most human and experimental tumor express higher than normal level of or abnormal forms of surface glycoprotein and glycolipids, including gangliosides, blood group antigens, and mucins. Tumor often have dysregulated expression of enzymes that synthesize the carbohydrate side chains of mucins, which lead to the appearance of tumor-specific epitope on the carbohydrate side chains or on the abnormally exposed polypeptide core. Several mucins have been the focus of diagnostics and therapeutic studies. One of these are mucin called MUC. Is an integral membrane protein that is associated is an integral membrane protein that is normally expressed only on the apical surface of the breast ductal epithelium, a site that is relatively sequenced from the immune system. In some carcinomas, however, MUC is expressed in non-polarized fashion and contain new tumor-specific carbohydrate and peptide epitope detectable by mouth monoclonal antibodies. Whether effective vaccine can be developed with these epitope remain an open question. Immune response to tumor. Both innate and adaptive immune response to tumor can be detected in patients and experimental animals, and various immune mechanisms can kill tumor cells in vitro. The challenges for tumor immunologists has been to determine which of these mechanisms may contribute significantly to protection against tumor and to develop therapy that enhance these effector mechanism in the ways that are tumor specific. Recent technical advance in characterizing tumor antigen specific immune response and data from the studies of cancer patients treated with a drug that it stimulates T cells have indicated that CTLS are the most important contributor to the host immune defense against tumor. In this section, we will review the evidence of anti-tumor immunity mediated by T cells and other immune effector mechanisms. T lymphocytes. The principal mechanism of immune protection against tumor is killing of tumor cell by CD8 CTLS. The ability of CTLS to provide effective anti-tumor immunity in vivo is clearly seen in animal experiments using carcinogen-induced and DNA viruses-induced tumor. CTLS or cytotoxic T lymphocyte may perform a surveillance function by recognizing and killing potentially malignant cells that express peptides that are derived from tumor antigen and are presented in association with class 1 MHC molecules. Tumor-specific cytotoxic T lymphocyte can be isolated from animal and human with established tumor and are discussed earlier. There is evidence that the prognosis for human tumor, including common types such as colon carcinoma, is more favorable when more cytotoxic T lymphocytes are present within the tumor. Furthermore, monoclonal cells derived from the inflammatory infiltrate in human solid tumor, called the tumor infiltrate lymphocytes, contain cytotoxic T lymphocyte with the capacity to kill the tumor from which they were isolated. Importantly, the inability to detect the function of tumor-specific cytotoxic T lymphocyte in some patients may be because of regulatory mechanism explored by tumor to suppress the CTL response. And new therapy that block this regulatory mechanism lead to the development of strong CTL response against the tumor, discussed later. CD8 T cells response specific for tumor antigen may require a cross-presentation of tumor antigen by dendritic cells. Most tumor cells are not derived from antigen-presenting cells and therefore are not present in secondary lymphoid organs where they can display antigen to naive T cells, nor do the tumor cell expresses costimulatory molecule needed for naive T cell activation. Thus, to initiate anti-tumor CD8 T cells response, tumor antigen have to be presented by dendritic cell, the best antigen presenting cell for transporting tumor antigen to secondary lymphoid organs and for activating naive T cell. Dendritic cells are the site of tumor. Dendritic cells at the site of the tumor can ingest tumor cells or their protein antigen, carry the tumor antigen to lymph nodes and colocalize with naive T cells. Furthermore, the dendritic cell can deliver the ingested protein from phagocytes into the cytosol so that they are processed by proteasome into peptides that are then displayed bound to class 1 molecule for recognition by CD8 T cells. This process of cross-presentation or cross-priming is mainly carried out by C dendritic cells, subset of dendritic cells. I described earlier in the chapter in the context of initiating CD8 T cell response to viruses. Dendritic cell also expresses co-stimulators, and these are helper T cells that are activated at the same time provide signal needed for differentiation of naive CD8 T cells in tumor-specific cytotoxic T lymphocyte. Once effector cytotoxic T lymphocytes are generated, they are able to recognize and kill the tumor cells without a requirement for co-stimulations. CD4 helper T cells contribute to anti-tumor immune response by several mechanisms. CD4 T cells response to tumor antigen are commonly found in animal model and patient with cancer, and the presence of T helper 1 cells like cytotoxic T lymphocyte in human tumor correlate with good prognosis. Some studies show a therapeutic benefit of adaptive transfer of tumor antigen specific CD4 T cells into the host. The anti-tumor effect of T helper 1 cells may reflect their own roles in enhancing CD8 T cells response and activating macrophage through the secretion of interferon gamma, as already seen in chapter 10. Interferon gamma can increase tumor cells class 1 MHC expression and sensitivity to lysis by cytotoxic T lymphocytes. The importance of interferon gamma in tumor immunity is demonstrated by the finding of increased incidence of tumor in knockout mice lacking these cytokines. Its receptor or interferon gamma inducing signaling molecule. So let us have a look on the diagram. So we are having cytotoxic lymphocyte response against tumor. Say tumor antigen are picked up by host dendritic cells and response are initiated in the secondary lymphoid organs. Tumor specific cytotoxic T lymphocyte migrate back to the tumor and kill tumor cells. CD4 T cells response against tumor involves similar initiating step to generate tumor-specific helper T cells, but anti-tumor effector mechanisms are different. Other mechanisms of tumor immunity are not shown. So in this, for the one who are going to see the diagram, we have tumor, and tumor, we see there is dendritic cells here, which are picking up some molecules from the tumor. Then in phagocytous tumors, they are taken back to the lymphoid. Then in activation of tumor antigen presenting CD8 T cells. Then after activating, then we have activated the T cells. The activated T cell migration of tumor cytotoxic T lymphocyte, then they migrate to the tumor sites, whereby cytotoxic T lymphocyte killing of tumor happens over there. So that is it. On figure 18.5, it say activation of tumor-specific CD8 T cells by cross-presentation. That is the diagram that is showing. Then a protein tumor antigen, or the cancer cell that produce the antigen, are ingested by dendritic cells into endocytic vesicle. The antigen are then delivered into the cytosols, where they enter the class 1 immunogenicity complex antigen processing and presentation pathway, resulting in the display of tumor peptide bound to class 1 MHC displayed on dendritic cells, along with costimulatory molecules. Naive CD8 T cells specific for these peptide MHC antigen can then be activated. So normally here you have tumor antigens, the cancer cell class MHC, then the antigen capture. After I've been captured, cancer cells tumor antigen picked up by antigen presenting cells, the class 2, then it will be processed, then it will be presented as class 2. Tumor antigen enter the cytosols. From the cytosol, then it can get into class MHC class 1, which will be presented to CD8 T cells. The demonstration that number of different types of T cells within resected tumor correlate with the likelihood of metastatic disease has led to the idea that determining an immune score for cancer may be useful to assess prognosis and direct treatment option. This has been mostly thoroughly studied in some medical center in cases of colon cancer, in which a score was given to tumor based on the number of CD45RO memory T cells and CD8 cytotoxic T lymphocytes in the margin of the resected tumor. A low score was found to predict a higher chance of relapse, metastasis, and death within 5 years compared with tumor with high score. Even when comparing tumor with no evidence of lymph nodes or distant metastasis at site of resection, in some study the immune score was found to have greater prognostic value than histological evaluation of tumor. Current research is focused on expanding the use of immune score for a wide range of tumor and broadening the analysis of resected tumor to include some subset of immune cells by immunohistochemistry and other methods. Additional immune inflammatory genes expression pattern for individual tumor are also being studied and may supplement immune score. Antibodies. Tumor-bearing hosts often produce antibody against various tumor. Tumor-bearing hosts often produce antibodies against various tumor antigens, but the significance of these antibodies in protecting against cancer is unknown. Antibody may kill tumor cells by activating complement or by antibody-dependent cell-mediated cytotoxicity, in which Fc receptor-bearing macrophage or natural killer cells mediate the killings. However, there is still a little evidence that humoral immune response against tumor has a significant effect in preventing the development and progression of tumor. There are also several approved and effective anti-tumor antibodies that are used to provide passive immunity against tumor, discussed later. Natural killer cells. Natural killer cells are capable of killing many types of tumor cells and may contribute to the immune surveillance against cancer. Some studies have indicated that people with defect in natural killer cells function or number caused by genetic mutation or with lower than normal natural killer cell activity without known genetic defect are at higher risk than general population of developing certain types of viral caused tumor. Mouse studies also have shown that genetic defect in natural killer cells function or depletion of natural killer cells by antibodies enhances tumor growth and metastasis. Although these findings support contribution of natural killer cells in immune surveillance, these cells usually represent only small fractions of inflammatory infiltrates present in most humans and mouse tumor, and their roles in immune eradication of established tumor is not clear. Tumor cells become susceptible to killing by natural killer cell when they down-regulate the expression of class 1 MSC, the appropriate expression of ligand that bind activating natural killer cell receptor. Natural killer cells expresses inhibitory receptor that bind to class 1 MSC molecules expressed on healthy cells. As we will see later, some tumor loses expression of class 1 MSC molecules as a result of the selection against class 1 MSC expression cells that are readily killed by cytotoxic lymphocyte. This loss of class 1 MSC molecule makes a tumor a particularly good target for natural killer cells. In addition, many tumor expressing ligands for NKG2D activating receptor on natural killer cells such as MYC-A, MYC-B, and ULB, and NKG2D signaling can override inhibitory signal from class 1 MSC binding receptor. Natural killer cells also may be activated to kill tumor cells coated with anti-tumor antibodies by antibody-dependent cell-mediated cytotoxicity. The tumoricidal capacity of natural killer cell is increased by cytokines, including interleukin-2, interleukin-15, interleukin-12, and the anti-tumor effect of these cytokines in vivo are partly attributable to stimulation of natural killer cell activities. Macrophage. Macrophage are capable of both inhibiting and promoting growth and spread of cancer depending on their activation state. Classically, activated M1 macrophage discussed in chapter 10 can kill many types of tumor. How macrophage are activated by tumor is not known. A possible mechanism is recognition of damage-associated molecule pattern from dying tumor cells by macrophage innate immune receptor. Macrophage in tumor also may be activated to kill tumor cell by interferon gamma produced by tumor-specific Th1 cells, cytotoxic T lymphocyte, and natural killer cells. This may be why a larger number of T helper 1 cells in some tumor is correlated with good prognosis. M1, which is macrophage, can kill tumor cells by mechanism that they also use to kill infectious organisms, including liberation of lysosomal enzymes, nitric oxide, and reactive oxygen species. We will discuss how M2 macrophages promote tumor growth in the next session. Role of innate and adaptive immunity in promoting tumor growth. Although much of the emphasis in tumor immunology has been the role of the immune system in eradicating tumor, it is clear that the immune system may also contribute to the growth of some solid tumor. In fact, chronic inflammation has long been recognized as the risk factor for the development of tumor in many different tissues, especially those affecting the chronic inflammatory disease such as Barrett esophagus, ulcerative colitis. Some cancers associated with infections are also considered to be an indirect result of tumor-promoting effect of the chronic inflammatory state that are induced by infectious organisms. These include gastric carcinoma and lymphoma in the setting of chronic Helicobacter pylori infection, hepatocellular carcinomas associated with chronic hepatitis B and C infection. Although the mechanism by which chronic inflammation can promote tumor development are not well understood, there are several possibilities supported by data in rodent model. Myeloid cells of innate immune system are considered the most direct tumor promoting culprit among immune cells. They may contribute to malignant transformation of cell by generating free radicals that cause DNA damage and lead to the mutation in tumor suppressor genes and oncogenes. Some data suggest that cells of the innate immune system, including mast cell, neutrophil, and macrophage, secrete soluble factor that promote cell cycle progression and survival of tumor cells. The transcription factor nuclear factor kappa beta, which is the key mediator of innate immune response, may play an important role in inflammation associated with cancer progression. Tumor-associated macrophage of alternative activated M2 phenotype, as well as other cells, are source of vesicular endothelial growth factor. Vesicular endothelial growth factor, a growth factor that promotes angiogenesis and matrix metalloproteinase enzyme that modify extracellular tissue. Therefore, chronic activation of some innate immune cells is characterized by angiogenesis and tissue remodeling, which favor tumor growth and spread. So we have the diagram here. Figure 8.6, promotion of tumor growth by anti-inflammatory tumor microenvironment. Although inflammation can promote malignant transformations of cell and the development of cancer, established tumor often create a microenvironment that suppress anti-tumor immunity and promote cancer cell growth. Tumor alter the phenotype of dendritic cell in the way that promote the differentiation of anti-inflammatory regulatory T cells and T helper 2 cells, which in turn promote differentiation and accumulation of M2 macrophage and myeloid-derived suppressor cells. So we have this is tumor-derived factor. This is tumor, the inhibitor tumor-specific effector T cells, myeloid-derived suppressor cells, differentiation of immunosuppressive myeloids. So all these M1 macrophage, these are actually the ones that works very well. But if you have a lot of myeloid-derived suppressor cells, they will prevent this to work. But apart from stimulation of tumor growth factor may cause this one to have angiogenesis and the blood vessels to get the tumor to get blood vessels, then it grows. Growth and androgenetic factors, tumor-associated M2 also macrophage, Th2 cells, cytokines, regulatory T cells, differentiation of anti-inflammatory cells. They can cause all this. So naive T cells, they cannot function that well, and therefore tumor progression is high. So these cells block the action of anti-tumor cytotoxic T lymphocytes, and Th2 cells provide growth factor for tumor cells and tumor blood vessel. The blue arrows show the changes of movement of cells. The black arrows show the effect of released factor from tumor in cells. So this is the diagram. Alternatively, activated macrophage and less well characterized cell populations such as myeloid-derived suppressor cells may also promote tumor growth indirectly by inhibiting effective anti-tumor immunity. The role of these suppressor cells immune evasion is discussed later. The adaptive immune system can enhance tumor development in several ways. In response to tumor, dendritic cell may be conditioned to drive CD4 T cell differentiation to T helper 2 cells or regulatory T cells Treg, both of which suppress T helper 1 and cytotoxic electrons that destroy tumor and increase the development of M2 macrophage and other cells that promote tumor growth. Experimental evidence in mice suggests that B lymphocytes may contribute to the tumor progression, but this remains controversial because B cells also have been linked to tumor regressions. The tumor promoting effects of immune system are paradoxical and topic of active investigation at present. A major problem is that the nature of the major tumor promoting immune stimulus vary among different tumor, and cell types that promote some tumors may inhibit others. As a result, it has been difficult to definitively establish the most important tumor promoting mechanism. These effects of chronic inflammation are theoretically target for pharmacological intervention because there is large variety of effective anti-tumor inflammatory drugs already available. The challenge of oncologists is to achieve a beneficial balance in which protective anti-tumor immune response are not compromised while potentially harmful tumor promoting inflammatory resection are controlled. While tumor promoting inflammatory reaction are controlled. Evasion of immune response by tumor. To grow in immunocompetent hosts, tumor have to evade or resist the host immune responses. Several mechanisms of immune evasion by tumor have been hypothesized and supported by experimental evidence or by clinical success of therapeutic approaches that target evasion mechanism. A major focus of tumor immunology is to understand this immune evasion mechanism with the goal that intervention to prevent immune evasion will increase the immunogenicity of tumor and maximize the effectiveness of the host response. Most evasion mechanism can be categorized as either active inhibition of anti-tumor immune response or loss of antigen that drive this response. So this is another diagram. So figure 18.7, mechanism by which tumor escape immune defense. Anti-tumor immunity develop when T cell recognize tumor antigen and are activated. Tumor cell may evade immune response by losing expression of antigen or major histocompatibility complex molecules by producing ligands for T cell inhibitory receptor and immunosuppressive cytokines. Tumor microenvironment, which include immunosuppressive myeloid cells and lymphocytes, inhibit the activation and differentiation and infiltration of anti-tumor effector T cells, cytotoxic T cells, and others, mostly cytotoxic T cells. So, anti-tumor immunity. Actually, tumor cells, tumor antigens, they will be shown by this one, by the antigen presenting cell. Then if recognize T cell recognition of tumor antigen leading to T cell activation. Production of immunosuppressive proteins or expression of inhibitory cell surface protein inhibitor. For example, PD1, PD ligand 1 from the antigen presenting cells will always inhibit the PD1 in T cells, therefore prevent killing. Immunosuppressive tumor microenvironment, like when you have a lot of Tregs, this can inhibit the process of T helper 1 and cytotoxic T cells, and also the other myeloid dystrophic system. Failure to produce tumor antigen, lack of T cell recognitions, then you cannot produce T cells, then they start growing. Lack of T cells recognition of the tumor also. Molecule and cell that inhibit immune response to tumor. Tumor evade anti-tumor T cells response by engaging T cell inhibitory immune checkpoint molecules that are normally function to prevent autoimmune or regulate immune response to microbes. This has been shown most clearly for CTLA, cytotoxic T lymphocyte associated protein 4, and PD1, programmed cell death 1, two of the best defined inhibitory pathway in T cells. The importance of these molecules in tumor immune evasion became clear from experimental work in mice showing that blockage of these inhibitors enhanced the anti-tumor immunity, followed by a successful clinical trial and now widely adopted use of anti-PD1 and anti-CTLA4 antibody to treat cancer. Tumors are able to resist T cell immune attack by upregulating the expression of PD1 and CTL4 tumor-specific T cell, probably as a result of chronic T stimulation by persistent tumor antigen. Tumor-infiltrating T cells often have a dysfunctional exhausted phenotype that was first described in the context of chronic viral infection. Exhausted T cells are characterized by impaired effector function and increased expression of CTL4, PD1, and other inhibitory molecules. In addition to upregulating CTL4 on tumor-specific effector T cells, the tumor's microenvironment may also promote differentiation of regulatory T cells which expresses CTL4. For PD1 and CTL4 to function as T cell inhibitor, they must bind their ligands. The ligands for PD1 are the B7 family proteins, PDL1, PD ligands 1, PD ligands 2. Many tumors expresses PD ligands 1 sometimes because of PD1 ligand gene amplification, and some tumor expresses PD ligands 2. Interferon gamma produced by tumor-infiltrating T cells also induces PD1 ligand expression on tumor cells and on the myeloid cells in the tumor microenvironment. Thus, many tumor or cell surrounding tumor can engage PD1 on infiltrating T cells and stop the T cell from performing their tumor killing function. The ligands for CTL4 are B71, B72, which are costimulatory molecule containing costimulatory molecule mainly expressing on antigen presenting cell in secondary lymphoid organ that work by engaging CD28. CTL4 bind to B7 more strongly than CD28 does, and thus act as competitive inhibitor for T cell costimulations. A possible reason for tumor can exploit CTLA4 to regulate anti-tumor responses that tumor antigen are often presented by antigen-presenting cell in the absence of strong innate immunity, and thus with low level of B7 costimulator on antigen-presenting cell. The low level may all be preferentially bound up to by the high affinity receptor CTL4 on effector T cells and Tregs, and therefore not accessible for CD28. In addition to PD1, CTL4, other inhibitory receptor expressed by tumor-specific T cells, including LAG3, TIM3, and TIGIT, also may contribute to the inhibition of anti-tumor immune responses. Secreted product of tumor cells may suppress anti-tumor immune response. An example of immunosuppressive tumor product is tumor growth factor, a transforming growth factor beta, which is secreted by many tumor and inhibits the proliferation and effector function of lymphocyte and macrophage. Treg may suppress T cell response to tumor. Evidence from mouse tumor studies and patients with cancer indicate that Treg can be found in cellular infiltrate in certain tumor. Depletion of Tregs in tumor-bearing mice enhances anti-tumor immunity and reduces tumor growth. However, the role and prognostic value of Treg present within human tumor remain uncertain and may vary among tumor cells. So, history of cancer immunotherapy. Some of the important discoveries in the field of cancer immunotherapy are summarized. BCG, Bacillus calmette-guerin, the chimeric antigen receptor, CTL, cytotoxic T lymphocyte, dendritic cell, FDA, PD1. So those are just explanation. So description of immune infiltrates by Vishal. That was 1863. Thereafter, treatment of cancer with bacteria products, colase toxins. That was 1898. Cancer immunosurveillance hypothesis, 1957. Treatment of bladder cancer with BCG, 1976. Interleukin-2 therapy for cancer, 1983. 1991-1994 discovery of human tumor antigens. 2002 adaptive T cell therapy. HPV vaccination to prevent uterine cervical cancer, 2009. 2010 FDA approval of dendritic cell vaccine Sipuleucel T for prostate cancer. Then 2011 FDA approval of anti-CTLA4 ipilimumab for melanoma. Then 2014 FDA approval of anti-PD1 for melanoma. FDA approval for anti-PD1 PDL for many cancer. It have been happening in 2016. So you can see this of the structure that how things have been happening. There have been many discovered in 2000s than in 19s, because there are very few discoveries actually. Myeloid-derived suppressor cells, MDSCS, are immature myeloid precursors that accumulate in bone marrow, lymphoid tissue, blood, and tumor-bearing animal and cancer patients suppress innate and T cell mediated anti-tumor immune response. Myeloid-derived suppressor cells are heterogeneous collection of different myeloid cell types, including cells that are most similar to monocyte, neutrophil, or dendritic cell. In addition to patients with tumor, MDSCC also accumulate in tissue of patients with chronic inflammatory diseases. Myeloid-derived suppressor cells are reported to suppress innate and adaptive immune response by many different mechanisms, including secretion of immunosuppressive cytokines such as interleukin-10, transforming growth factor beta, promotion of Treg differentiation, and inhibition of cytotoxic T lymphocyte and T helper 1 differentiation. Cyclooxygenase 2-dependent production of prostaglandin E2 may enhance differentiation of myeloid-derived suppressor cells. With this immunosuppressive function. Although the presence of MDSC in tumor correlates with impaired anti-tumor immune response, there are many gaps in our knowledge about the nature of these cells, how they develop and function, and how they can be targeted for therapeutic purpose. As mentioned earlier, M2 macrophage activated by tumor may inhibit anti-tumor immunity and promote tumor growth. Loss of tumor antigen expression. Immune response to tumor cells impart selective pressure that result in the survival and the outgrowth of variant tumor cells with reduced immunogenicity. Experiment comparing tumor that develop in normal mice versus RAG deficient mice that lack adaptive immune show that only in the setting of normal immune system do tumor become less immunogenic over time, which is consistent with selective survival of less immunogenic clones. This phenomenon has been called immune editing, implying that the immune response selected for changes in tumor that help them evade the response. Given the high mitotic rate of tumor cells and their genetic instability, mutation and deletion in gene encoding tumor antigen are common. If these antigens are not required for growth of tumor, or maintenance of the transformed phenotype, the antigen-negative tumor cell will have growth advantage in face of host immune response. Recent studies have confirmed this occur in cancer patients. Tumor-specific antigen that drive T-cell response in the patient were identified by full exome sequencing and identification of mutant peptide that found to the patient's MSC allele. In those patients, tumor subclonal could be detected that no longer carried the mutation that encode immunogenic neoantigens. Class 1 MSC expression is often downregulated on tumor cells so they cannot present any tumor antigen to CD8 T cells. This is likely a more effective form of immune evasion than mutation loss of individual antigen. Whereas tumors show decreased synthesis of class 1 MSC molecules or protein required for class 1 MSC expression on cell surface such as B microglobulin or component of antigen processing machinery such as transporter associated with antigen processing TAP1, TAP2 subunit of protosome. Loss of expression of class 1 MSC molecules or proteins involved in MSC assembly and antigen presentation are due to mutation in the gene encoding these molecules. Tumor cells with such mutation presumably have a selective survival advantage because they can evade CTL-mediated immune response. As we discussed earlier, tumors that lose the class 1 MSC are more likely to be recognized by natural killer cells. However, additional mutation that impair tumor cells' expression of ligand natural killer cells activating receptor may emerge, promoting outgrowth of subclonal that also evade natural killer cells attack. Immunotherapy for tumors. Oncologists and immunologists have worked for many years on the immunological approach to treat patients with cancer, and only recently have been exciting and broadly applicable breakthrough that have been successfully used to treat patients. A major reason for interest in immunological treatments is that most established therapies for cancer relies on drug, chemotherapy, or radiation that kill dividing cells or block cell division, and these treatments have harmful effect on normal proliferating cells. As a result, the treatment of cancer causes significant morbidity and mortality. Immune response to tumor can theoretically be highly specific for tumor cell and will not injure most normal cells. Therefore, immunotherapy has the potential for being the most tumor-specific treatment that can be devised. Recent advances in identifying the tumor antigens and method for genetically modifying T cells so that they are specific for those antigens have brought us closer to tumor-specific immunotherapy. The breakthrough approach now is in practice that stimulate the immune response to control tumor are not entirely tumor antigen specific and do have side effect of damaging normal tissue. Nevertheless, these approaches provide great benefit to many patients. I repeat, the breakthrough approach now in practice that stimulate the immune response to control tumor are not entirely tumor antigen specific and do have side effect of damaging normal tissue. Nevertheless, these approaches provide great benefit to many patients. So these are some of the drugs and things that are used. Monoclonal antibody approved for cancer therapy. We have specific for antibody drugs name. For example, for HER2 or new EGFR, we have trastuzumab. Humanized. For treating breast cancer, we have CD19. For example, blinatumomab, which is for acute lymphoblastic leukemia and cell-mediated tumor killing. We have CD20 antibody, which is called the rituximab, and is for treatment of T-cell lymphomas and leukemias, chronic lymphocytic leukemia. CD20 again, for example, we have the one which is 90Y ibrutomab Tuxtan for low-grade transformed B-cell non-Hodgkin lymphoma. CD30, which is called brentuximab vedotin for Hodgkin or systematic anaphylaxis large cell lymphoma. CD33, it is called gemtuzumab. The other one is oxaligammaxins. These are for acute myelogenous leukemia. CD52, alemtuzumab for CTL, for LLL, CTL, and the T cells lymphoma. We have EGFR, for example, cetuximab pantino for the colorectal cancer breast. We have VEG, we have CD54. This one you can study yourself. A second major reason to explore immunological approach for treating tumor is that cytotoxic drugs have been unsuccessful in achieving durable benefit in most cancers that have spread in the body beyond the site of origin, because long-lasting memory is the cardinal features of adaptive immune response, and immunity is systemic. It is possible that once an effective adaptive immune response to tumor is initiated, it will be sustained for a long time and it will be effective throughout the body. Because this feature of the immune response, there is a hope that some immunotherapeutic approach will achieve long-term cure. In this section, we will describe different modes of tumor immunotherapy that are currently used clinically or in development. Passive immunotherapy with monoclonal antibodies or antibody-like molecules. Passive antibody therapy involves the transfer of specific antibodies into patients, which is a rapid and theoretically very specific approach but does not lead to long-lived immunity. Some monoclonal antibodies have been used to treat cancer for over 20 years. We have table 18.1. Furthermore, recombinant single polypeptide proteins with the antibody-like antigens binding site called the single-chain variable fragment specific for tumor antigen have been developed for cancer treatment. Some anti-tumor antibody bind to cell surface molecule on tumor cell and engage host effect mechanisms that kill the tumor cells. These mechanisms include natural killer cell mediated cytotoxicity, complement-mediated lysis and complement Fc receptor-mediated phagocytosis by macrophage. Several anti-tumor antibodies that are now approved for treatment of certain cancer work in this way. For example, as mentioned earlier, anti-CD20 is used to treat B-cell lymphoma or lymphoma, and it works by depleting all CD20 expressing cells, including B cells and B-cell derived lymphoma cells, mainly antibody-dependent cellular cytotoxics, and perhaps also complement activation. Immunotoxins or conjugated monoclonal antibodies are antibodies specific for tumor antigen that are linked to chemotherapy drug and or radioisotopes. The rationale for this agent is they will allow high local concentration of cytotoxic drugs or isotopes to be delivered to the tumor cells. Because of the antibody specificity, several drugs conjugated antibody are approved for clinical use, including those specific for HER2/neu to treat breast cancer, CD30 to treat Hodgkin lymphoma, CD23 to treat acute myeloid leukemia, CD22 to treat some acute lymphoblastic leukemia, CD79B to treat some B lymphocyte lymphomas. Many more antibody drugs conjugate have been developed, but some have failed in clinical trial because of significant systemic toxicity due to non-specific non-specific accumulation of the toxic component in various tissues. Other monoclonal antibodies used in cancer therapy bind to growth factor receptor in cancer cells and interfere with the signaling required for tumor growth and survival. Anti-HER2/neu is approved for monoclonal antibody used to treat breast cancer that expresses the cell's surface growth factor signaling molecule HER2, and the antibody that bind and block the functioning of epidermal growth factor receptor is approved for treatment of metastatic colorectal cancer and head and neck cancer. Another antibody in clinical use for several cancer block are endothelial growth factor, VEG, that stimulate the angiogenesis that is required to maintain tumor growth. In this case, antibody does not bind to tumor cells. Bispecific B cell engagers BiTEs. Bispecific T cell engagers facilitate the target of the host T cells of any specificity to attack tumor cells. These reagents are recombinant protein engineered to express two different antigens binding site, one specific for tumor antigen and the other second specific for T cells surface molecule, usually CD3. In BiTEs, each antigen binding site is composed of a single chain variable fragments, which is a single polypeptide containing tandem immunoglobulin, heavy and light chain variable domains. The presumed mechanism of action of BiTEs, which is bispecific T cell engagers, the presumed mechanism is based on vitro studies in the formation of immune synapses between the tumor cells and T cells and the activation of T cells by CD3 ligations. A CD19 specific BiTEs is approved for treatment of acute lymphocytic leukemia. BiTEs specific for many other tumor antigens have been developed, including CD20, BCMA, EpCAM, HER2, EGFR, CA, folate receptor, CD33, and are at various stages of preclinical development and clinical trials. Adaptive cellular therapy with the anti-tumor T cells. Adaptive cellular immunotherapy is the transfer of immune cells that have anti-tumor reactivity into a tumor-bearing host. The immune cells that are derived from are derived from a cancer patient's blood or solid tumor and then are treated in various ways in vitro to expand their number and enhance their anti-tumor activities before reinfusion back to the patients. Sorry, we forgot to pass through this diagram. So actually this is cancer patients who receive these different types of management. These are mechanism of action of anti-tumor antibodies. Many different monoclonal antibodies that bind directly to a molecule on the surface of cancer cells are used as drug to treat cancers. Some of the main mechanisms by which these antibodies work to kill cancer cells are shown. Other monoclonal antibody used for cancer immunotherapy that target soluble growth factor or cell surface molecule on immune cells are not shown. So antibody specific for T cells, tumor antigens, tumor antigens. So they have the antibody, they activate the complement system, they can kill the tumor. Antibody specific for cell surface tumor internalizations, and it can be killed in many other functions, including the CTLs. So we are here at adaptive cellular therapy with autologous tumor-specific T cells. T cell specificity for tumor antigen can be harvested from a patient tumor tissue or blood and expanded and activated in vitro and infused back into a cancer patients. This general approach has been used in various trials for many years, but has limited success, probably because the cells that are isolated from the patient contain low frequency of potent tumor-specific T cells. With the advent of the technology discussed earlier to identify neoantigens that drive tumor-specific T cells response in individual patients, there is a renewed hope for adaptive therapy with T cells specific for these antigens. The approach will involve harvesting the T cell from the blood or tumor of the patient, stimulating the cells with tumor neoantigen in vitro to increase the number and functional activities of cells specific for the tumor, and then transferring the activated T cell back into the patients. Thereby, there have already been some success with a small trial using this approach in patients with melanoma. Another T cells adaptive therapy approach currently in development is transduce a patient's T cells to express T cell receptor specific for tumor antigen commonly expressed by a particular tumor types and displayed by a known HLA human leukocyte antigen molecule. This gene encoding the T cell receptor polypeptide chains are derived from cloned T cells taken from a patient with that tumor and are incorporated into lentiviral expression vector that can be used to infect T cells taken from the patient with that tumor and with the same HLA. This approach will require identifying antigens that are target for T cells and expressed in the same type of tumor in different patients. T cell receptor genes may be optimized by introducing mutations that enhance the affinity of T cells to the antigens. Chimeric antigen receptor T cell therapy. Adaptive therapies using T cells expressing chimeric antigen receptor CARs have proved successful in some hematological malignancies, and this approach is being developed for other tumors. CARs are genetically engineered membrane-bound receptor with a tumor antigen specific binding site encoded by recombinant Ig variable genes, a single chain variable fragment, and cytoplasmic tails containing signaling domain of both the T cell receptor and T cells costimulatory receptor. The reason for using an anti-tumor body like binding site as tumor antigen recognition receptor is that this approach avoid the problem of MSC restriction of T cell receptors. So the same CARs construct can be used for a particular tumor types in any patient. In addition, CARs will be able to recognize tumor antigen even if tumors stop expressing MSC molecules, which is a frequent mechanism of immune evasion. Genetically engineered cytoplasmic tail for CARs contain signaling domain that normally serve critical roles in T cell activation. Several variations of signaling constructs have been used so far in CARs development in different centers, but all contain the T cell receptor zeta chains immunoreceptor tyrosine based activation motif and cytoplasmic signaling motif of costimulatory receptor such as CD28 and CD137 401BB. These signaling motifs center on the tumor's Ig-like receptor, the ability to potently activate T cells. In current protocol, a cancer patient's peripheral blood T cell are isolated, infected with CAR encoding retroviral and lentiviral vector, and then stimulated with anti-CD3, anti-CD28 antibodies to expand their number. The expanded CAR-expressed T cells are then injected back into the patients. The prior transfer of the patient are usually treated with drug that deplete their own lymphocyte, which maximize the proliferation of the transferred CAR T cells. The transferred T cells undergo robust expansion in the patients in response to tumor antigen recognition by the Ig domain, by the CAR and costimulatory signal providing by signaling domains. The specificities of endogenous T cell receptor on these T cells, which are still present, become irrelevant to a goal of killing tumor cells because all the CAR cell expressing T cell can be activated by tumor antigen that bind to receptor encoded by CAR genes. Tumor killing is achieved by both direct cytotoxic and cytokine mediated mechanism. CAR T cell specific for CD19, a pan B cells marker expressed on B cell derived tumor cell, is approved for treatment of virus B cells malignancies that are refractory to other treatment, including chronic lymphocytic leukemia, acute lymphoblastic leukemia, and B cells lymphomas. And CD20 specificity CAR cells are approved for B cells lymphomas. Memory CAR T cells may persist in the treated patients so that surveillance against tumor recurrence is maintained. The technologies to produce large number of CAR T cells for each patient in a short time have advanced greatly, and CAR T cells are now used in many medical centers. So let us read a little bit about the diagram. It says adaptive T cell therapy. T cells isolated from the blood of the cancer patients activated in vitro to expand their number and induce effector phenotypes, and then infused back to circulations of the same patients. Some of these T cells that are specific for tumor antigens will be activated by and kill the tumor cells expressing those antigens. So that is number one. B is chimeric antigens receptor, CARs, a genetically engineered membrane-bound receptor that recognize tumor antigen by antibody like binding site and generate intracellular signaling that activate T cells. CAR T cells are generated by transducing a cancer patient blood T cells into vitro with viruses engineered to express the CARs and are expanded into vitro before infusion back to the patient. CAR cells have been successful in treatment of certain leukemias and lymphomas. So this was the diagram actually. So the diagram, see here you have T cell removed from the cancer patients, you have taken it, then you have T cell activated and expanded in vitro, you have expansion, then you transfer back, then after transfer back, then this one, tumor antigen specific T cells, they fight against tumor and kill tumor. But another way is adaptive transfer of chimeric antigen receptor, CAR-Ts. So here T cell removed from cancer patients' blood, then T cell activated and expanded in vitro with anti-CD4 and then expansion, the T cell infected and antivirus express tumor antigens, CAR genes. Then after give again to the patient's CAR T cell mediated killing tumor cells happens. So there remains some significant roadblock that will need to be overcome for successful expansion of use of CAR T cell therapy. The tumor antigen targeted by CAR T cells may be expressed by some normal cells, which also will be killed. In the case of the first CARs in clinical use, specific for CD19, normal B cells are killed, but if necessary, patient can be supplemented with pooled Ig to make up the lack of B cells. Because long-lived antibody-producing plasma cells found in adult bone marrow and mucosa do not express CD19 and are not killed, they continue to provide antibody-mediated immunity in adult patients treated with CD19 specific CARs cells. Another problem commonly encountered in CARS2 cell therapy is cytokine release syndrome, a dangerous adverse reaction that frequently occurs soon after adaptive transfer of T cells into patients with a high tumor burden. In those patients, so many of the T cells become activated at the same time that an intense systemic inflammatory response occurs due to cytokines secreted by T cells, which in turn stimulate cytokine release from macrophages and other cells. Some patients who develop this reaction have been successfully treated with the anti-interleukin 6 receptor antibody. The severity for this complication can be decreased by treating patients with the cytotoxic chemotherapy to reduce tumor burden before CAR cells transfer. This limits the magnitude of CAR T cell activation that occur often after transfer. Other patients have developed serious neurotoxicity after CAR T cell infusion, which may be related to the microvascular injuries or the effect of secreted cytokines that get into the brain, and risk of the long-term damage to the central nervous system remain concerned, especially in children whose brain are incompletely developed. So we have the chimeric antigen receptor here. The chimeric antigen receptor CARs are composed of extracellular immunoglobulin single chains variable fragments specific for tumor antigen and cytoplasmic signaling domains that activate T cells, such as zeta chains immunotherapy, tyrosine-based activated motif items, and motif in the cytoplasmic tail of the costimulatory receptor CD2841BB, which promote robust T cell activations. So we have tumor cells, tumor antigens, then items activate the patient T cells, then killing mechanism happen. Tumor may lose expression of antigen being targeted by CARs and recur. The selection pressure imposed by CARs T cells likely promote the emergence of antigen tumor variant. This problem may be reduced by simultaneously expressing CAR specific for two or theoretically more tumor antigen. The chances of tumor clone losing multiple antigen is less. In some patients, transferred CAR2 cells appear to become unresponsive over time, and the initially controlled tumor have recurred. The CAR cells in these patients expresses markers of exhaustions, including high level of PD-1, and resembles the exhausted T cells mentioned earlier. This problem can be addressed by treating the CAR2 T cells recipients with blocking antibodies specific for PD-1, or by using genome editing method to eliminate the PD-1 genes in the CAR T cells to avoid the risk of autoimmunity induced by PD-1 negative T cells. Endogenous T cells are also eliminated from a T cell before transfer. This creates T cells that have only introduced tumor-specific antigens receptor and do not become exhausted because they lack immune checkpoint molecule. Attempts to treat solid non-hematological tumor with CAR2 cells have been hampered by difficulties in identifying optimal antigen target for CAR2 cells on solid tumors that are not also expressed on the normal cells whose destruction would cause serious toxicity. The reason for this may be that the cell surface antigen which have been the target for CARs2 are usually not tumor specific and are often differentiation antigen or signal receptor that are expressed at some level or normal cells of particular lineage. One approach to overcome this problem is to identify pair of antigens that are commonly expressed together only on tumor cells and to transduce T cells with two different CARs, each specific for one of the antigen and each with different signaling domain, both of which have to be engaged to activate T cells. It is hoped that antigen can be identified such that the likelihood of tumor cells expressing both antigen is greater than that of any normal cells expressing both. Getting the injected CAR cells to migrate to the site of solid tumor is also a challenge. Strategy to genetically engineer the CAR T cell with the enhanced migratory function are in development. Studies are also ongoing to express CARs in cells other than CD8 T cells, such as natural killer cell, with hope that these will be effective in killing tumor but will cause less toxicity than CAR T cells. Immune checkpoint blockage targeting T cells inhibitory pathway. Blockage of T cell inhibitory molecule has emerged as one of the most promising methods for effective enhance the patient's immune response to their tumor. This approach is based on the idea that tumor cells exploit various normal pathways of immune regulation or tolerance to evade the host immune response as discussed earlier. Because this inhibitory mechanism establishes checkpoint in the immune response, the approach of stimulating immune response by a drug that inhibits the inhibitors is called immune checkpoint blockade. The first drug developed in this class is a monoclonal antibody specific for CTL4, the receptor on T cells that bind B71B72 and block T cell stimulation, especially during T cell priming in secondary lymphoid organs. Anti-CTL4 was first approved as therapy for advanced melanoma, and it is effective in stopping or slowing tumor progression in many, but not a majority of treated patients. This antibody may work by blocking the action of CTL4 expressed on activated T cells and on Treg. As discussed earlier, T cells' response against tumor also may be inhibited by PD-1 ligands, PD-1 pathway, which works by activating a phosphate that blocks the activation of effector T cells. Antibody blockage of PD-1 or its ligand appear to be even more effective than anti-CTL4 in enhancing T cell killing of tumor and halting the progression of otherwise lethal advanced cancer. Anti-PD-1 and anti-PD-1 ligand antibody also cause less severe adverse effect described later than does anti-CTL4, and these antibodies are now approved for treatment of many types of cancer including melanoma, lung carcinoma, renal carcinoma, bladder carcinoma, colon carcinoma, Hodgkin lymphoma, and others. In fact, anti-PD-1 is approved for all recurrent and metastatic tumor with the mismatch repair defect, which result in high level of mutations and thus production of abundant neoantigen in every pathologic types of tumor. This is the first cancer therapy that has been approved based on a genetic signature of the tumor, regardless of the tissue or cells of origin of the tumor. It is likely that the anti-tumor T cells that respond to this type of therapy in each patient are CD8 T cells that recognize a neoantigen-derived peptide presented by class 1 MHC molecules. Combined blockage of both PD-1 and CTL4 appear to be more effective against certain cancer than either alone and is approved for several cancers. So we see the diagram here. This is figure. The checkpoint blockade. Tumor patients are often mount ineffective T cell response to their tumor because of upregulation of inhibitory receptor such as CTL4 and PD-1 on the tumor-specific T cells and expression of the LIGO PD-L1 on the tumor cells. Blocking CTLA4 antibodies, anti-PD-1, anti-PD-L1 antibodies, and B are highly effective in treating several types of advanced tumor by releasing inhibitory tumor-specific T cell by these molecules. Anti-CTLA4 may work by blocking CTL4 on responding T cells or regulating T cells, CTLA4 cytotoxic T lymphocytes, and others. So here we have the diagram. Induction of anti-tumor immune response in lymph nodes. So we have tumor peptide, we have T cell receptor. This is the dendritic cells, but we have B7, we have CTL4, and we have this CTL4 and this CTL4 and B1. They have to come together, and it was supposed, this CD28 was supposed to interact with B7, but because of inhibitory no co-stimulation, therefore tumor cells cannot be killed. But in co-stimulation it can happen if it happened that there is anti-CTLA which can block this the CTLA. Then the CD28 is free to interact with the normal ligands which is used, the normal ligand which is used, and we can mention easily there as we see. So then primary CTL capable of killing the tumor cells. In B, CTL mediated killing tumor cells, tumor peptides here. As we know, we have to remember in our mind that CTL is mostly available in CTL, in CD8 T cells and other, and also PD-1 may be there. So normally this one is PD-1 ligands can be expressed, then this one can prevent and inhibit CTL to happen. But if you have PD-1 antibodies, they will bind PD-1L and bind this one, therefore leave the free chance for this one to interact and work to dead tumor. So we go. Common adverse effect of checkpoint blockage treatment for cancer, autoimmune and inflammatory reactions, collectively called immune-related adverse event, which is predictable in light of known roles of CTLA4 and PD-1 in maintaining self-tolerance and regulatory T cell response. The most frequent adverse events involve inflammations of colon, lung, liver, and various endocrine organs. Although many other organs and tissue, including muscle, the heart, and central nervous system, can be affected, some of these inflammatory disorders are unusual in absence of checkpoint blockage, such as autoimmune destruction of anterior pituitary glands, acute onset and rapid destruction of pancreatic islet, leading to insulin-dependent diabetes. Most of these adverse events can be successfully treated can be successfully treated with anti-inflammatory medications such as corticosteroid and or corrected with hormone replacement therapy, but many require cessation of checkpoint blockage therapy, and some, such as myocarditis, have high mortality rates. Overall, only 15 to 20% of the patients with cancer treated with the CTLA4 or anti-PD-1 have objective improvement in their disease, and among these, some tumor recur after initial response. There are several possible reasons for this therapy failure. The first one, checkpoint blockage therapy is unlikely to work in patients with tumor that have relatively few somatic mutation encoding neoantigen, because there will be few clones of tumor-specific T cell that will respond. Many tumors do not take advantage of CTLA4 and PD-1 pathway as strategy to evade anti-tumor immunity, but rather employ other immune evasion mechanisms consistent with concept of low level of expression of PD-1 on some tumor types detected by immunohistochemistry predicting poor response to anti-PD-1 therapy. PD-1 ligands expressed tumor that initially respond to anti-PD-1 therapy may become resistant in the presence of strong immune response. The acquired resistance could occur by selective growth of clones of tumor cells that express molecular other than PD-1 that inhibit T cells response. Alternatively, clones of tumor cells may be selected that induce the T cell to express other checkpoint receptor beside PD-1. An important goal of cancer immunologists and oncologists is to identify histologic or genetic feature of tumor or circulating biomarker that may predict which patient will respond best to which checkpoint blockage therapy. The nature of the cellular infiltrate around the tumor has some predictive values of the response to checkpoint blockage. In general, abundant effector T cells, even if they have a phenotype of dysfunctional or exhausted cells, predict a good response, where sparse cellular infiltrates or an abundance of Treg predict poor response. In the future, assays for T cell expressing antigen receptor specific on neoantigen may be combined with analysis of neoantigens abundance and provide greater predictive value. The presence of B cells in tumor infiltrate also correlates with good response to checkpoint blockage. It is not known if this is because B cells and antibody contribute to anti-tumor immunity, or if the presence of B cells implies a robust immune response against the tumor. To increase the percentage of patients who respond to checkpoint blockage, oncologists are testing the efficacy of blocking other T cells inhibitory molecules including LAG, TIG, and TIM3, alone or in combination with anti-PD-1s, anti-CTL4. Other approaches including combined checkpoint blockage with more conventional radiations or chemotherapy protocol, combining checkpoint blockage with tumor vaccine discussed later. Kinase inhibitor that block oncogenic pathway in the tumor or stimulating agonistic antibodies specific for activating receptor on T cells. Vaccination with tumor antigen. Vaccination of the tumor-bearing individual with tumor antigen may result in enhanced immune response against the tumor. The earliest attempt to boost the anti-tumor immunity relied on non-specific immune stimulation. More recently, vaccine composed of killed tumor cells, recombinant tumor antigens, or dendritic cell incubated with tumor antigen have been tested in animal model and in clinical trial with patients with cancer. The identification of peptide recognized by tumor-specific CTL and the identification of mutant genes that encode tumor-specific antigens recognized by CTL have provided many candidate antigen to include in tumor vaccines. New DNA sequencing technology are now widely used to rapidly determine all the mutations in protein-coding DNA sequence exomes of cancer cell genomes. Prediction algorithms are applied to these data to identify the mutant peptide that are most likely to bind to the MHC allele of each patient. This technical advances now allow for the precise identification of tumor-specific neoantigens in individual tumor, and this has stimulated efforts for development of personalized tumor vaccines. Tumor vaccination strategies employ a variety of adjuvant and delivery methods. Pro-inflammatory molecules are used to enhance number of activated dendritic cells at the vaccination site. These adjuvants include toll-like receptor ligands such as CpG, DNA, and mimicry of double-stranded RNA, and cytokines such as granulocyte, macrophage, colony-stimulating factors. Tumor antigen are delivered in the form of dendritic cell vaccine, dendritic cell vaccines. In this approach, dendritic cells are purified from the patient, incubated with the tumor antigens, and then injected back into the patient. A dendritic cell-based vaccine is now approved to treat the advanced prostate cancer, but it has not proved to be effective in most patients. Technical challenges with dendritic cell vaccine are that cells have to be harvested from each patient, and they require expansion in cell culture, which is difficult to standardize. Sure. DNA vaccines and viral vector encoded tumor antigens are being tested in clinical trial. This may be the best way to induce CTL, cytotoxic lymphocyte response, because the encoded antigens are synthesized in the cytosol of cells such as the dendritic cell and efficiently enter the class 1 MHC class pathway of antigen presentation. Overall, the result of trial with many different types of tumor vaccines have been inconsistent and generally not very successful. Unlike many standard vaccines against microbes with the prophylactically prevent infection, tumor vaccines are used as therapy that need to be effective to stopping progression of tumor that have already developed. Most tumor vaccines tried to date have targeted antigens that are commonly shared by same tumor types in different patients, and these antigens are usually differentiation antigens and also expressed on cells of the normal tissue from which the cancer arose. Vaccine using such antigen have generally not been successful, likely because the antigen in normal cells induce tolerance that has to be overcome of induction of effective anti-tumor immunity. The development of virus-induced tumor can be reduced by preventive vaccination with viral antigen or attenuated live viruses. As mentioned earlier, HPV vaccines have been effective in decreasing incidence of HPV-induced premalignant lesion and cancer in the cervix. This approach has also been extremely successful in reducing the incidence of failing leukemia virus-induced hematological cancer in cats and preventing market disease, a herpes virus-induced lymphoma in chickens. So, detecting tumor neoantigen that elicit T-cell response. Tumor DNA can be purified, and exome sequence can detect random mutation in genomes of cancer cells. A computer algorithm then can be used to determine which mutation occur in amino acid sequence that encode peptide that could bind to the major histocompatibility complex allele in that patients. The validity of putative neoantigens peptide can be tested for assay of patient T cells response to these peptide in vitro or testing if the MHC peptide multimeric complex can bind to the T cells. This approach is being used to create personalized tumor vaccines. So here, if someone has tumor, it is taken and cultured, then we obtain DNA. Next generation sequencing identify tumor specific mutations, then identification of the mutated HLA binding peptide by computational algorithm, identify T cells from patients that are specific for putative neoantigen by functional assay binding MSC peptides. So you get some cells which will be directly able to kill that. So here a figure 8.14 dendritic cell vaccine. Dendritic cells generated in vitro from blood monocyte taken from the patient with tumor can be pulsed with a defined tumor antigen and infused back to the patient, where they will present with antigen to T cells specific for antigen and boost the tumor specific immune response. In other approach, the dendritic cells are transferred with genes encoding the tumor antigen, sometimes also cytokines that promote immune response, and these cells are used as vaccines. Other approaches for stimulating anti-tumor immunity. Several additional approaches have been used to enhance host immunity against tumor, with variable success. Cytokine therapy. Cancer patients can be treated with cytokine that stimulate the proliferation and differentiation of T lymphocytes and natural killer cells. These cytokines can enhance activation of dendritic cell and tumor-specific T cells, particularly CD8 cytotoxic T lymphocytes. Many cytokines also have potential to induce non-specific inflammatory response, which by themselves may have anti-tumor activities. The largest clinical experience is with a high dose of interleukin-2 given intravenously, which has been effective in inducing measurable tumor regression in about 10% of patients with advanced melanoma and renal cell carcinoma, and is currently an approved therapy for this cancer. The use of high-dose interleukin-2 is, however, limited because it stimulates the production of toxic amount of pro-inflammatory cytokines such as tumor necrosis factor interferon gamma, which act on vesicular endothelial cells and other cells and lead to the serious vesicular leak syndrome. Interferon gamma is approved for treatment of several cancers, including malignant melanoma, certain lymphomas, and leukemias, AIDS-related Kaposi sarcoma. The mechanism of antineoplastic effect of interferon gamma probably include inhibition of tumor cell proliferations, increasing cytotoxic activity of natural killer cell, increasing class 1 MHC expression on tumor cell, which makes them more susceptible to be killed by cytotoxic T cells. Other cytokines such as tumor necrosis factor interferon gamma are effective anti-tumor agents in animal model, but their use in patients is limited by their toxic side effects. Hematopoietic growth factor, including GM-CSF and GCF-CF, are used in cancer treatment protocol to shorten period of neutropenia and thrombocytopenia after therapy or autologous bone marrow transplantation. Oncolytic viruses. Oncolytic viruses are genetically modified viruses that selectively replicate in and cause lytic death of cancer cells, and in doing so promote cytotoxic T lymphocytes response specific for tumor antigen that are released. The first oncolytic virus approved for clinical use is talimonogens laheroparvec (TVEC) to treat metastatic melanoma. TVEC is herpes simplex virus in which genes have been deleted to enhance viral-induced cell lysis and reduce chronic infection of nerves. Furthermore, a gene that encodes for TAP inhibitor has been deleted. thus enhances the class 1 antigen presentation pathway in infected cells, and gene encoding GM-CSF has been added, which increases the dendritic cell accumulation in tumor microenvironment. TVEC is injected directly into solid tumor site, but enhances anti-tumor cytotoxic T lymphocyte response even at distance location from injected site, which is the evidence that oncolytic viruses promote systemic anti-tumor process. Anti-tumor immunity. Clinical trials are being conducted to test for the efficacy of TVEC on other tumor types, and other oncolytic viruses are in development. Non-specific inflammatory stimuli. Immune response to tumor may be stimulated by the local administration of inflammatory substance, or by systemic treatment with agents that function as polyclonal activator of lymphocyte. One of the oldest examples of tumor immunotherapy was practiced by 19th century physician William Coley, who treated his cancer patient with extract of dead bacteria, so-called Coley's toxin. This approach may be seen intermittently successful due to their induction of strong innate response leading to production of tumor necrosis factor and other cytokines that cause acute inflammation that kill the tumor cells. Non-specific immune stimulation of patient with tumor by injection of inflammatory substance such as kill the Bacillus calmette guerin at site of tumor growth has been used in many years. The BCG mycobacterium activate macrophage and thereby promote macrophage-mediated killing of tumor cells. In addition, bacteria function as adjuvants and may stimulate T cell response to tumor antigens. Intravesicular BCG is currently used to treat bladder cancer. Cytokine therapy discussed earlier represents another method of enhancing immune response to non-specific manner. Graft versus leukemia effects. In leukemia patients treated with the allogenic hematopoietic stem cells or transplant, the presence of T cells and natural killer cells in hematopoietic stem cell inoculum can contribute to the eradication of tumor. The T cells mediated graft versus leukemia effect is directed at molecule present on the recipient's hematopoietic cells, including leukemia cells that are recognized as foreign by administration of T cells. Donor natural killer cells respond to the tumor cell because tumor may express low level of class 1 MSC molecules, or they express a class 1 MSC allele not recognized by donor natural killer cells. Recall that recognition of self class 1 MSC normally inhibits the activation of natural killer cells. The challenge is in use of this treatment to improve clinical outcome. The challenge in use of this treatment to improve clinical outcome is to minimize the dangerous graft versus host disease that may be mediated by same donor T cells. The remarkable recent advances in cancer immunotherapy promise to dramatically change the care of the patient with these dreaded diseases. The success of checkpoint blockage for many solid tumors and of CAR T cell infusion for hematological malignancies has revitalized the field of tumor immunology. Although limitation problem remains, the enormous effort being invested in this field makes it likely that further advances will happen rapidly. Summary. Tumor expresses antigens that are recognized by immune system, but most tumor suppress immune response or weak immunogenic and immune response often fail to prevent the growth of the tumor. Nevertheless, the immune system can be therapeutically stimulated to effectively kill tumor. Tumor antigens recognized by cytotoxic T lymphocytes are the principal inducer of and target of anti-tumor immunity. Tumor-specific neoantigen generated by random mutations of cellular proteins, which can be processed into the major histocompatibility complex binding mutant peptides, are the most important. But other tumor antigens known to stimulate host cells include product of mutated oncogenes, normal protein whose expression is dysregulated or increased in tumor, and antigens of oncogenic viruses. Antibodies specific for tumor cells antigen are used for diagnosis, and the antigen are potential target of antibody therapy. These antigens include oncofetal antigen, which are expressed normally during fetal life and whose expression is dysregulated in some tumor. Altered surface glycoprotein and glycolipids and molecules are normally expressed on cells from which the tumor arises are thus differentiation antigen for particular cell types. Immune response that are capable of killing tumor cells are mediated by cytotoxic T lymphocyte, natural killer cells, and macrophage, possibly activated by tumor antigen-specific helper T cells. Among these immune effector mechanism, the role of CTL in protecting individual from tumor is best defined. Tumor evade immune response by several mechanisms, including downregulating expression of MHC molecules, selective growth of cells that do not express tumor antigens, production by soluble immunosuppressive substance, the engagement of inhibitor receptor on lymphocyte by the ligand expressed on the tumor cells, and induction of regulatory T cells. Tumor-associated macrophage and myeloid-derived suppressor cells found in most solid tumor can suppress anti-tumor immunity. Immunotherapy of tumor include approaches that augment active immune response against these tumor, provision of tumor-specific immune effector to provide passive immunity to patients. Anti-tumor immunity may be enhanced by blocking mechanism of immunoregulatory. Immune response also may be actively stimulated by vaccination with tumor or antigen and by systemic administration of cytokine that stimulate immune response. Anti-tumor antibodies are used widely in tumor immunotherapy. The antibodies bind to the molecules on the surface of tumor cell and engage effector mechanism to kill the tumors, including complements. Natural killer cell and phagocytes or antibodies bind to growth factor receptors, which block the signaling needed to sustain tumor growth. Tumor antigen-specific antibodies also have been conjugated with the chemotherapeutic toxins or radioisotopes to target these agents specifically special to tumor. Some genetically engineered biospecific antibodies simultaneously bind the tumor antigen and activate receptor for T cells. CAR T therapy is a form of cancer therapy in which a patient T cells are engineered ex vivo to express hybrid antigen receptor, chimeric antigen receptor, that recognize the tumor antigen by antibody V domains and signal via cytoplasmic T cell receptor and the costimulatory receptor motif. The CAR cells are then transferred back to the tumor cell patients, thereby where they become activated in tumor antigens and kill the tumor cells. CAR therapy has been effective in treating some hematopoietic tumors. Immune checkpoint blockade is the mode of tumor immunotherapy in which function blocking antibodies against the inhibitor receptors on T cells or their ligands, including PD-1, programmed cell death 1, programmed ligands, and CTLA, cytotoxic lymphocyte antigen 4, cytotoxic T lymphocyte antigen 4, are administered to remove the brakes on lymphocyte activation and thus promote anti-tumor immunity. Previously inhibited host T cells specifically for antigen. Checkpoint blockers have been widely adopted to treat many types of cancer. So what follows are selected readings that you can go on and read. Thank you for your time again. This was Jeremiah Puza.